we've done a GWAS, and so everyone's familiar with that. And so to date, there have been tens of millions of individuals who've been included in large-scale genome-wide association studies as well as in sequencing studies for identifying genetic variants that contribute to variation of complex traits. So the vast majority of these studies, including all the Alzheimer's disease studies, have been conducted in populations of European ancestry. So Carlos Bustamante and his group in 2011 did a survey of all the GWAS that have been reported in the literature and found that 96% focus on European populations and 4% in non-European populations. So a huge bias, sampling bias for these studies. Okay? So last year, Alice Pope-Joy, who's a PhD student at UW, and Malia Fullerton at UW as well, they were looking at the current state of affairs and looking at the genetic studies and seeing has it changed in the past 7 years. And surprisingly found that there was some improvement where 81% of the GWAS sequence studies were conducted in European samples. But what's somewhat surprising, too, is that with this improvement, 19% in non-European, but the vast majority of those were conducted in China, in Asian populations. So we still had a large number of individuals who were excluded, particularly underrepresented minority populations, such as Hispanics and African Americans. And so just to get a better idea of this, the majority were Asians, and so of the other individuals who were excluded largely were the African Americans, Hispanic, Pacific Islanders, and other populations. Okay, so we still have this huge imbalance in the samples that are being used for identifying genetic variants for complex traits. So bias understanding which variants are important. So we know, for example, heritability. We know heritability, for example, is population specific. Certain variants are important for certain traits in certain populations than other variants. So there are certain variants that are shared across populations. Some variants are population specific, and heritability is population specific. Okay, so you have certain traits that have a higher prevalence in one population than the other, and a lot of that has to do with the frequency of the causal variants differs from population to populations. Okay, so this has impact potentially for healthcare inequalities. And so we really need genetic studies, genomic studies in diverse populations. Okay, so as I mentioned, medical genomics has focused almost entirely on those of European ancestry. Okay, other race and ethnic groups must be studied to ensure that more people benefit. So I always like to have talks like this to really motivate why I think this is really important. Okay, so we're in this era now of personalized medicine where essentially, we're trying to identify which individuals would benefit from certain drug treatments. So we know certain people respond to certain drugs and do well, other people don't. So what are some of the factors that contribute to this? Okay, so you give certain people drug treatment. It's an effective treatment for some. It's not effective for others, but in some segment of the population, it actually is harmful. Okay, so what are some of the factors that contribute to this? Okay, so I'm going to give this asthma example with one of my collaborators at UCSF, Esteban Richard, where asthma affects about 334 million people globally. Okay, the prevalence of asthma, what do you say? Puerto Ricans, yeah. The prevalence of asthma is highest in Puerto Ricans. Okay, African Americans is next, and then Caucasian, then Mexicans. For some reason, Mexicans, for example, have the lowest prevalence, which still is baffling. Okay, you look at mortality. Okay, Puerto Ricans almost 4 times that of Europeans. Okay, if you look at the health disparities, clearly you see that individuals who would need the greatest benefit potentially are the Puerto Ricans. Okay, 4 times mortality, and so albuterol is really the only medication that's prescribed. Oh, you got it. Okay. You want to give this talk? You want to give this talk?
And it's interesting to note the question just came up next to me.
Okay.
That also the risk does not change when people move.
Yeah.
It's really
in Puerto Rico. Exactly. Yes. So with Puerto Ricans, so albuterol is the most prescribed medication. So do you know Esteban? You should talk with him. Okay. So he's been leading the genetic of asthma and Latin American study, and he's been recruiting children and looking at drug responses to albuterol, okay, based upon racial and ethnic groups, and found something pretty shocking. One is that for Puerto Ricans, okay, it's not very effective at all. So really, you need to be here to have,
so much money.
yeah. Okay. African Americans the same way. So my son has asthma. He puffs all day and it does nothing for him. We battled our school system and say we're not paying for this medication anymore because it's not doing anything. Okay? And so these are some of the issues, is that the drugs, for example, that have been prescribed for this condition that affects Puerto Ricans disproportionate to other population is the least effective in them. Okay? The good news for the Mexicans is that it's not effective for them either, but they have a lower prevalence of it. Okay? So even more depressing is that for those who take albuterol, African Americans who, for example, and it does not work, they're often prescribed ÇMCA. Okay? Now this is medication that is on the market now, that's used widely. Okay? There's a black box warning in this long list that says in African Americans, asthma-related deaths occurred at a higher rate in patients with ÇMCA than those treated with placebo, 7.26 times higher than a placebo, than doing nothing at all. Okay? This is medication that's on the market right now. It's approved. Okay? So,
did they know that at the time it was approved, or was it that found out?
Yeah. I got it. I mean, so, they do it in clinical trials based on Europeans mostly,
Yeah,
right?
exactly.
So I think people started dying, and then they go back, and now they have put the black, it's still out there, but they got the black box warning.
Yeah.
But if you pull this, if you had prescriptions, this long list, it's tiny, the little bit that you see there. Okay, so it's scary. So these are some of the things that we're trying to understand why. Why are African Americans, for example, 7 times more likely to die taking this medication than nothing at all? These are people who suffer from asthma, so they need to be taking something, but they're better off not taking anything at all. Okay? So we're in this era of the precision medicine initiative. So in 2015, Obama, initiated the precision medicine initiative, and he said it gives us one of the greatest opportunities for new medical breakthroughs that we have ever seen. Okay. I wonder if that's going to be around in the next 2 years, since everything's getting reversed. Okay. So we have the Precision Medicine Initiative that was launched by the NIH. So the goal is to have a cohort of more than 1 million or more Americans. And we want to support advance of targeted prevention and treatment, precision medicine, taking into account individuals' unique characteristics, including individual genome sequences, environmental factors, and lifestyle. So one of the components of the Precision Medicine Initiative is the Top Med program. Has anyone heard of Top Med yet? Okay. So it's ongoing. It's the Transomics for Precision Medicine. So basically, whole genome sequencing data is being generated for more than 120,000 individuals. Okay. So more than 30 cohorts with well-defined phenotypes and existing clinical outcomes data are included in this. So I'm sure you guys analyzed some of this data. Framingham is part of this. Jackson Heart Study, MESA. So a lot of the studies that I'm sure you're aware of, they're part of Top Med, where they're getting their samples and we're getting whole genome sequence data on these individuals. Deep sequencing too. Okay, so the primary aims is to identify genetic variants for increased or decreased risk of disease, as well as those that help disease subtypes. So we talked about GWAS. GWAS were very useful for identifying common variants that may be involved with traits of interest, but with sequence data, we can get the rare variants that potentially may explain some of the missing heritability. Okay. So to date, about 70,000 whole genome sequences have been completed. And so I'm involved in this project because UW in Biostat, we're the genetic analysis center. So we're getting all the data, we're processing it, and we're helping with the analysis with this data. But what I'm really excited about this is that it's very diverse. Okay, so at least the first phase of this, where we have about 20,000 samples, about half are European, 30% African, 10% Hispanic, Latino, 8% Asian, but still Pacific Islanders were still grossly underrepresented. But it's still much better than the chart I gave you earlier. So we have a much greater diversity in top med. Okay. So hopefully I've motivated why it's important to do genetic studies in diverse populations. And so there's tremendous opportunities to identify potentially novel genetic variants that underline phenotypic diversity between populations. Okay. So also potential to provide new insights into health disparities. Can we identify what are the genetic variants that are involved in why, for example, Puerto Ricans have the highest prevalence of mortality of asthma than Europeans, for example, and Mexicans as well. But there are some serious challenges for complex trait mapping in diverse populations. Okay? One of the biggest challenges, heterogeneous genetic backgrounds. Okay, how do you account for that? Confounding due to ancestry. I'm going to talk about that in a minute, population structure. How do you infer it, and how do you correct for it? Okay? And finally, potentially dealing with familiar and cryptic relatedness, but I'm going to talk about confounding and population structure today, as that's what Paul asked me to talk about. Okay? So at its most basic, case control association study, you did a GWAS, you just did association testing at every single marker. Essentially what you're doing, if you have a case control phenotype, okay, either you have disease or you don't, Alzheimer's disease or not, okay, if you have a binary trait, is you're comparing allele frequencies in the cases to the controls. So here's an example. I have cases and controls. I'm looking at a genetic marker. I'm going to arbitrarily label them as having alleles big A and little a. I can either compare the genotype frequencies, okay, and say, I'm seeing a frequency of the big A genotype more than I'm seeing in the controls, or you can compare, for example, alleles. Most of the time we do some type of, assuming some additive model, and we're really looking at the number of copies of alleles. So I can look at the allele frequency of the big A alleles higher in the case in the controls, and then we want to know, is this difference that we see due to just random chance, or actually the A is really associated with case control status. So feel free to stop me if anything isn't clear. Okay. So one of the biggest concerns in genetic association studies are false positives. Okay, you're doing a lot of testing, trying to get signals, hundreds of thousands, maybe even millions of markers. Okay, and so it's well known that ancestry differences among individuals can cause spurious association of false positive results. Okay, so population structure is another way to talk about ancestry differences. It's just the presence of subgroups of the population with ancestry differences. So if you neglect that, or you fail to account for it in your analysis, you can have spurious association. So it's a serious concern, okay, in genetic association studies. So for the epidemiologists who like these types of graphs, essentially we're looking at, we want to know if genotype or genetic variant is associated with my trait of interest. Okay? So a confounding variable is a variable that's associated with both your outcome of interest and your predictor, okay, but it's not in your causal pathway of interest. Okay? We want to know, is genotype associated with my trait, but many traits of interest are associated with ancestry. Okay? So if I'm looking for an association between genotype and trait, I want to know, is association between genotype and trait, is it direct association but not through ancestry, okay, if that makes sense. Okay? So many traits of interest are correlated with ancestry because ethnic groups and subgroups share environments, environmental and genetic variation, dietary habits, and other lifestyle characteristics. Okay? And so if you don't take into account ancestry differences, you can pick up all genetic variants across the genomes that's telling me that these people have different allele frequencies based on race. Okay? So if you don't account for ancestry differences, you'll get a bunch of spear association in diverse populations. So you have to account for that properly. So this is an example. If in population A, for a case control study, population A is in red and population 2 is in blue, 1 and 2, for example. If I oversample the cases in red and undersample the blue cases, and then I have a balance, for example, number of cases and controls from population 1 to population 2, then I'm going to get overrepresentation among cases, and then genetic markers that are not involved with my disease but have significant differences between the populations are going to show up. Okay, so those are spear associations.
So can you walk us through how that might present a problem specifically for Alzheimer's disease research in African Americans, where the African Americans who show up to clinics and are more likely to participate in ADME or in NASH, any Alzheimer's research center, are more likely to be cases.
Okay.
Because then the whites who present to clinic. And the controls are usually from a completely different, the controls among African Americans are completely different. So in other words, how might that, what you've done there in your sample
Yes.
is you've oversampled from the cases from A and perhaps undersampled them from B. And
Exactly.
I think that can definitely happen.
Yeah.
If you're doing a clinic-based sample.
Yeah.
For Alzheimer's disease, looking at ethnic minorities compared to whites.
Yeah. So it happens all the time. Okay. So like I said, so we have many situations where and this population structure can be subtle. I'll talk about that in a minute. But what would happen, for example, if I have more Africans here that are cases, let's say I analyze the Africans and the Europeans together. Okay, and I have more Africans samples here and half and half here, but I'm overrepresenting the Africans in the cases. Don't forget that in a case control study, all I'm really doing is comparing the allele frequency between cases and controls. So any genetic variant where the allele frequency is higher or lower in the cases than in the controls, I'm picking up, regardless of whether or not it's involved with a trait of interest. So I'm going to get any ancestry informative marker where the allele frequency is different in Africans and controls. That's going to show up because ancestry is hopelessly confounded with my case control status. So I'm going to pick up all genetic variants where I have a higher frequency in cases, for example, than in controls, and it has nothing to do with my trait of interest.
So that can happen if you're overdiagnosing Alzheimer's disease in African Americans because your tests are inappropriate.
So we're going to talk about how to infer it and how to account for it, though. So we have ways to adjust for it in our analysis. Okay, but
is it the same problem as overdiagnosing, isn't it? Or do you mean overdiagnosing, like misdiagnosing? Yeah, misdiagnosing. Is it the same problem, though? Isn't it slightly different?
Like if you think of the extreme, if you get all African Americans as your cases and all whites as your controls,
Right.
then you would pick up all of the genetic differences between African Americans as whites, and you would say, oh, that's the case. That's the cause of AD, but it's really not. It just has to do with how different.
But that's so regardless of what your study design is. So even if you've oversampled the cases from population A and controls, we want to account for that. So regardless of the study design, we can account for those differences so that we don't have spurious association. But we have to good inference on these differences in ancestry among individuals if ancestry is a confounder. So sorry if it's not clear, but we can talk a little bit more later.
So when you're talking about ancestry, you're talking about
Genetic ancestry.
And so how do you define somebody falling in African American story as a certain percentage of their
That's a good.
ancestry that has to be African? Like where's the cutoff?
There's no cutoff. So we get. I mean, in the U.S., if you had an ounce of black blood, you are African American. In the U.S., that was the rule, right? So we need to infer that. So we have some, for example, some of the studies I'm involved with, we have some African Americans who look almost 100% European, right? And so we have some African Americans who look like they're straight off the boat from Ghana, right? So we have this range, and so we need to infer that. So we don't want to rely on self-reported ancestry. Not to say that self-reported ancestry isn't important, right? So in an example, for many traits of interest, for example, there's a non-genetic component, for example, where culture, diet can impact a lot of these traits of interest. You want to account for that. So we saw, for example, we had some African Caribbeans in top med, and we had African Americans. For some of these traits, the African Caribbeans look very different than the African Americans. But look at the admixture ancestry; they look very similar. But for BMI and things of nature, that has a lot to do with diet and culture and what they're eating as well. Okay, so not to say it's not important, but if I'm talking about confounding due to genetics, I'm talking about inferring their ancestry from genetic data. And so we could talk about estimating it using ancestry proportions. There are methods there. We're going to talk about that a little bit later. But also we're going to talk about other ways where we don't have to really put them in groups. Like, yes, exactly. Okay. So even if you have, question? Yeah, it's not AD. Yeah. So we're going to talk about admixture in a minute. Yeah, that's right. Yeah, exactly. Yeah, that's right. Yeah. So this is true for quantitative traits as well. So here, for example, if population, the blue population has a higher value of a trait. For example, African Americans Europeans have higher white blood cell count than Europeans. We know there's a Duffy gene that's involved with low white blood cell count in African Americans. If you look at the distribution of African Americans, it's much lower. If you don't take that into account, you're going to get, and you have Europeans and Africans in your sample, you can get a lot of variants that are predictive of ancestry but have nothing to do with the trait of interest, unless you adjust for it or account for it appropriately. Okay? So it's not just with case control studies, it's also with quantitative traits. Here's for top med for BMI. Okay? So we analyzed the self-reported African Americans, the white, and the white includes like the Amish as well, and others. We accounted for relatedness, and we looked at some of our results for our QQ plots. If you do something like Plink in this study, it's not going to work. You need more sophisticated tools to account for the relatedness and population structure. Plink is a nice tool for doing simple analysis, but for more complex analysis, there's other software for that. So here, for example, we're looking at BMI. If you look at the African-American distribution, it's very different than the self-reported white. Okay? So we have many traits of that where quantitative case control studies, we talk about the asthma with the Puerto Ricans and everything like that. Sorry, man. Yeah, I mean, that's my go-to example. I've never had a Puerto Rican in my talk, so yeah. Great. Yeah, exactly. So, and I should point out that quantile normalization won't save you here. So even if you do quantile normalization and you try to make things normal, you still have this distribution that differs by race and ancestry. Okay? So it's a serious problem, and we're still coming up with tools to handle it. Okay? So population structures, little background. Okay, so while accepted that original humans were in Africa, and then we had the out of Africa, where now we have individuals from different populations. Migration, genetic drift leads to genetic variation between isolated populations. Okay, so due to genetic drift, we have individuals who have different ancestries, okay, and different genetic profiles. Okay, so inferring genetic ancestry differences among individuals with different populations has been motivated by a variety of applications. Okay, so population genetics. So a lot of people do population genetics, looking at population genetic history, genetic association studies, which most people here are interested in, personalized medicine, okay, what I talked about, okay, and even in forensics. So there's some issues with forensics when the FBI is trying to identify, individuals who are criminals based upon the STRs that they identify. Like it doesn't work so well on other populations that they're using. Okay, so there's some issues even there with forensics. Okay, what are the appropriate models that we can use to match DNA based upon a person's ancestry? Okay. But in any case, advancements in array-based and sequencing technologies have really facilitated this field of inferring genetic ancestry, and we're now able to investigate it at remarkably high level of detail. So there have been many methods available that have been developed to identify genetic ancestry differences among individuals in a sample using genome screen data. By far, the most commonly used approach is principal components analysis. Has everyone heard of PCA? Done it? Okay. I see some no's and some yeses. You'll be doing one today in the session in the afternoon. But PCA, principal components analysis, is applied to genotype data. Okay? And so we calculate the principal components that best explains this variation that we see in our genetic data, where the top principal components are viewed as continuous axes of variation that should be reflecting ancestry in a sample, where along a similar component, hopefully the component is reflecting some ancestry component, people should have similar ancestry along a certain axis. So it should be noted that PCA is unsupervised. Okay, so this is where it's an agnostic approach. Okay, so we're not making any assumption about which ancestries are in there. We let the data make, we plot the data and we see where individuals cluster. Okay. But it's widely used in the genetics community to infer ancestry differences, with the premise being that the top PCs, top principal components, will reflect population structure. Okay. So principal components identifies orthogonal axes of variations, and they're just essentially linear combinations of the SNPs that should be explaining the majority of the variation in the data, or the greatest proportion of variability, and these define the PCs. Okay? So the Eigensoft implementation is, from Alkes Price's paper, really was often used, but now we have a lot of tools in R. So I have to put equations in there. My apologies in advance, okay? But hopefully it'll be clear. So you guys have GWAS data. You have data across the genome. So let's say you have a sample of N individuals. Okay, so how many people were in the ADNI data set?
What the one we used
Today, 691. So let's say N is 691. How many markers did you have? About 500,000. Okay, so in here, N could be, for ADNI data, 691. The number of markers is 500,000. Okay, so at each marker, we have a genotype value. So we're going to code the genotype values as 0, 1, or 2. You choose some reference allele, it does not matter. If we're coding it as additive, it doesn't matter what the reference allele is. It could be the minor allele or the major allele. And so for each individual at a marker, the individual has a value 0, 1, or 2. Right? So we want to standardize everybody's genotype. So why are we standardizing, first of all? So what is this doing? So to standardize every genotype, I'm subtracting the mean value in a sample, okay, 2 times the frequency divided by 2b1 minus p. So first, what is that doing right there? Any thoughts? Why are we standardizing? So we're standardizing every genetic marker. So why are we doing that? So on the same scale. Okay. So they have essentially on average mean 0 and variance 1. So now I'm comparing individuals or markers that have different allele frequencies. Now I've standardized them, so now I can compare these markers across the genome so they're on the same scale with mean 0 and variance 1. Okay, so I've essentially scaled my data and standardized it. So, question? Uh-huh.
This came up this morning in our group. Now, the way I understand principal components, M is much bigger than N.
Yes.
So you get a variance-covariance matrix that is singular.
Yes, but yes and no, because we get a correlation matrix. So for all possible pairs of individuals, so for example, we have a matrix that essentially the IJ matrix is a correlation matrix. So what I do essentially, I get the correlation for all possible pairs across the genome. So at the end, I'm going to end up with an N by N matrix, right? Where each element in the matrix is basically an average correlation of the markers.
So it's going to be N by N.
N by N, number of samples. So for the ijth individual, I average the correlation of those markers across the genome. So I have a correlation matrix n by n. Okay? So for all possible pairs, I have a correlation. Okay? And it's a correlation in some sense because we standardize it, right? Because all our values are standardized. So the idea is individuals who have similar ancestry, what should their correlation be? It should be some positive correlation. People who have more different or divergent ancestry should have some negative correlation. So this measure really tells us how genetically similar our samples are. If that makes sense. So this comes up a lot. So you'll see this in the literature a lot. A genetic relatedness matrix, genetic similarity matrix, is essentially a measure of how correlated everyone's genome is, but we're averaging across the genome for all possible pairs. Okay. So now I have my matrix. Okay, I can look at my matrix and say, okay, which individuals are correlated, okay, which individuals are not. I have my correlation matrix, it's symmetric. Now, if I have a matrix, okay, for the math folks here, I can get an eigen decomposition of that matrix. So all matrices can be written in terms of the eigenvectors and the eigenvalues. Okay, so all symmetric matrices can be written in this way, where you have some vectors and then you have some diagonal elements of the eigenvalues. Okay, so don't worry too much about it, but just note that the most important eigenvector is going to be, of course, is going to be the first one. The less important one is going to be the second, and so forth. But the most important eigenvector that's explaining most of the variability in the data should be the first one, followed by the second. So these are eigenvalues, eigenvectors. Okay. So just note that we can decompose our similarity matrix into this form where the most important vectors that are explaining most of the variation in the data is going to be the first one, then the second one, and so forth. Okay? So the top principal components are the top eigenvectors. So they explain most of the variation. So that variation that we see in the data should be due to people who have similar different ancestries. So where people who have similar ancestry should be clustering together, okay, on the top PCs. People who have different ancestry should be separated from each other. Okay. So the top principal components are viewed as continuous axis variation. So the top principal component corresponds to the top eigenvector. The top eigenvector has the eigenvalue that's the top. So don't worry too much about it, but if you want details, this really explains it. But the idea is individuals with similar ancestry along a certain axis should have similar values for their principal component. So how many do you use? So you're going to get n principal components.
Right.
Okay. It's going to depend on your sample, right? So some people, some studies you only may need one. Other studies you're going to need many more. Like for top med, we got such diverse samples. Some of our studies we have more than 10, particularly if you want to get some more of the subtle structure. Yeah, so it's going to depend. And a lot of times you're going to have to do some diagnostics of that. Right? You're going to do some plotting of the principal components and see if I'm still seeing some type of strange patterns. Okay. Okay. So what about principal components? How did this really become popular? So there was a paper by John Novembre in Nature in 2008 where he did PCA in Europeans where he only included individuals in the study who had both grandparents born in the same country in Europe, used 200,000 SNPs, and matched their country of origin. Okay? And interestingly, he found that the top PC, two PCs, nicely mirrored the geography. Okay? So this was a seminal paper. They no doubt helped John win the Genius Award for this. Yeah. So this is a major paper. Okay?
Isn't that cool?
Yeah. It's scary too. Yeah. That they can pinpoint from your genetic data? Yeah, that's, I don't know. I mean. Yeah. Yeah. Okay. And so what did they do in this study? They had their samples. They got a genetic relatedness matrix, looked at all possible pairs. They got their genetic similarity matrix, got the top PCs, and plotted the first two. Okay? And showed that it nicely mirrored geography.
What does it mean that, so the purple, I assume, is Spain?
So this is Spain and Portugal. So these are the countries of origin that these individuals are from.
So what does it mean that the orange is so different in the spatial coordinates than, say, the yellow for France? Does that mean that Spain doesn't really mix with France that much?
No. So they only included samples whose both grandparents were from that country. Yeah. So here, I mean, you do have some individuals from Spain that cluster in with the French here, but you do see a nice pattern. But it's not necessarily so clean, but there is a nice pattern, right? Like you really can't distinguish Portugal from Spain very well.
Yeah, it just looks like Spain is so different from England, well, from France.
Yeah,
In
I
terms
think
of this
space. Yeah, I don't. Yeah. So how to interpret it and why, I don't know. But it was amazing that it even matched this the way it did, as well as it did. It corresponded. Okay. Question.
Yeah, I was just wondering, what are the caveats with that? I mean, the data collection and how it was used. Like, does it actually mean that you're from there? Or
No, right?
And so, I mean, just talk about some of the caveats, or maybe are you going into that?
So no, does it mean so? It was a reason why they only included, for example, individuals that have been there for 3 generations. I think they're more likely to have have genetic clusters. But I mean, as you see, you got some individuals from Spain who are up here, and so how to interpret that? I mean, like if you give me a totally different sample, the pattern should be somewhat similar, but people can cluster with other individuals. So what would you interpret about, for example, the French that are down here? Somehow maybe their ancestors came down there from France down to Spain years ago. I don't know. It's hard to interpret what that means. But once again, this is unsupervised, and their genotypes cluster with the majority of the Spanish and the Portuguese individuals. So I think that's pretty much what we can say, is that their genotypes are really more similar in their clustering.
And it was all self-identified.
Self-identified, but both grandparents had to have come from.
Both your grandparents, like for me, it would be both my grandparents were German.
Yes.
And then they only stuck with Germans.
Yeah, so these should be somewhat representative of 3 generations of Spain. Now, beyond that, what does that mean? But yeah, exactly. Self-identify, yeah. Yeah, exactly. Question?
I'll just add, I remember the first time I saw this, that at least my own interpretation of it was that this really gives weight to this idea of kind of old school population drift, right? That migration started in Africa as people moved to different places at different times, and bottlenecks happened, and selection happened, and so we ended up with these genetic variations that exist across the globe, and that we can still in that almost exactly information state, which is kind of amazing. That the sort of, to me, it just gave weight to the reasons why we need to take population stratification very seriously when we do this kind of thing.
Yeah, agreed. Thank you.
And just to reinforce the idea, if you happen to enroll more cases who happen to be from Northern Europe and a smaller proportion of cases from Southern Europe, and that's just the way it happens to be, any of these genes that's associated with that north-south axis will show up as associated with case status, even though they're completely about where your grandparents are from.
Yeah.
Right? And not that you're due to case. So by throwing in the principal components, you then are controlling for the north-south axis in Europe and the east-west axis in Europe.
Yeah, exactly.
And it looks really slick.
Yeah.
They must have had coordinates of the country as part of the principal component.
No.
It's just genetic data.
There may be some orientation or something, but no. This is agnostic. They colored after the points.
Yeah, but principal components can be rotated.
Yeah. It may be some orientation.
They've done some jiggling
to make it. Yeah, they might have done some transformation to make it. Yeah, exactly. But like I said, the PCs, they were done agnostic. There was no information about Ancestrally. It was unsupervised. So that's within Europe. But there can also be population structure even in populations that seem to be relatively homogeneous. So there was a nice paper written in 2009 where they looked at population structure within Finland. So even really subtle structure within Finland, they're showing that there's a nice relationship between the top two PCs. So this is similar to PCs, this PC analysis, but you see there is some pattern there as well, even within Finland. Okay? So basic idea, someone mentioned about man-to-mate, people are more likely to mate with people who are similar to them, live near them, okay, or have similar ancestral backgrounds. And so there really is no random mating. Okay? So even with subtle structure, you can pick some of that up with PCA. Okay. So you have your PCs. You're trying to do your association analysis. So Price et al. Proposed to correct for confounding due to population structure by including the PCs in a regression model. Okay. So you're interested in association between your SNP, I'm going to call my SNP X, with 0, 1, and 2 copies. You include PCs in your model as surrogates for ancestry. Okay. So now, by including PCs in the model, what is that essentially doing? It's saying that we're adjusting for baseline differences in my outcome that are due to ancestry. So now when I'm talking about is there any association with my trait, I'm really only comparing individuals who have the same ancestry. So I'm looking for mean differences in my outcome after adjusting for ancestry. So I'm looking for mean difference in my outcome among individuals who have the same ancestry but have different genotypic values. So that's essentially what including.
Each of those PCs is basically a linear combination of all of the SNPs that you're analyzing?
Yeah, so PCs are linear combination sets, but they're orthogonal. The PCs are completely orthogonal, number one. And number two, you can find out which SNPs are driving the PC. So likely there's only going to be a few SNPs that are driving, for example, PC1 that are really telling us about the east-west European cline, for example. Okay.
So that's what we did today, actually. We had all that PCA.
Yes, exactly. Yeah. So Joey, you did PCA with the ADNI data. Yeah. So that's essentially what you did. Okay. Okay. So a little warning that these PCA approaches that have been proposed really assume that individuals are unrelated or distantly related. We're all related to some point, right? You go far back enough, we're all going to have a common ancestor. Okay, so if you do PCA on family samples, okay, your top PCs may be clustering people who are family because it's just telling you about genetic relatedness. So essentially, distinguishing familial relatedness from ancestry using genotype data is a difficult problem that we've been focused on. So my former PhD student had a nice paper, Matt Kahnemo's, really showing how to do robust inference of population structure when you hold up relatedness and population structure. So we want to separate similarity in genetics that due to familial relatedness, parent, offspring, siblings, from genetic similarity due to people who share a more distant common ancestor. So Matt actually won the best paper award for this work. So if you do have related samples, definitely check out this PCA program that can give you inference on ancestry in the presence of close relatedness. Okay. So there's a question about admixed populations. Okay. So a number of large-scale studies include populations that are admixed. So these are populations characterized by ancestry derived by two or more ancestral populations that were previously isolated. So they've risen in the past few hundred years as a result of historical events such as the slave trade, the colonization of the Americas, and other long-distance migration. African Americans are admixed, largely with European and African ancestry. Hispanics, that's a total different. They're admixed. It depends on what type, if you're Puerto Ricans or Colombians or Puerto Ricans or Dominicans. Very different admixture. We'll talk about that a little bit later. Cape Verdeans in South Africa. So there are a lot of populations who have diverse ancestry from continental populations that were previously isolated. So if you look at the genome of an admixed individual, for example, if you had two populations, for example, European and Native American, colonization of the Americas, these genomes came together. And then now if you look at the individuals who are admixed, their chromosomes are essentially mosaics of the ancestral population. So if we actually could look and look at the chromosome, there'll be a chunk from Europe and a chunk from Native America and a chunk from Africa. So hopefully someone knows this, who's not a geneticist. Why don't we have entire chromosomes here? Why do we have like a mosaic? What's the process for why we have European, then chunks of Native American, for example, and then chunks of European? What is that process? Recombination. So after one generation, when this individual mates, one of these two chromosomes combines and gets passed down, it's going to be a mosaic, so because of recombination. And so actually, because of recombination, there are a lot of people focus on, based upon the number of combination and how long the ancestral haplotypes are, they're trying to figure out like the time of admixture. So there's a lot of work going on with that as well. So even in admixed populations, there'll be substantial heterogeneity. Admixed populations have diverse ancestry and are therefore structured. They have population structure. So there are methods available to actually estimate their proportional ancestry instead of PCA, but you can use a supervised approach. So you can estimate proportional ancestry from the ancestral populations. So methods such as admixture are supervised, where you essentially give them genetic data from ancestral populations, for example, that are surrogates, European, African American, and then you give an individual admixture genotype data, and it kind of figures out, we'll talk about that in a minute, what proportion of ancestry that individual is from these different continental groups. There are other methods that can actually estimate precisely at a particular genetic marker. So you have SNP data, 0, 1, 2 copies of a reference allele. With these methods like RF-mix, you can actually estimate whether the allele came from Africa or European ancestry or Native American or so forth. So we can actually get to the level of determining, for an admixed individual, the population of origin of that allele. And so there's another field called admixture mapping, where instead of testing for an association of 0, 1, 2 copies of the allele, it's tested for association of 0, 1, 2 copies of your ancestry at that marker. So that's another topic. So local ancestry we can estimate, and we can also estimate genome-wide ancestry. So I'll talk about genome-wide ancestry very quickly, but essentially we have an admixed individual. What it tries to do is you have an individual who has this DNA, and it tries to match the DNA to some reference population panels, 1,000 genomes or HapMap. And then based upon the proportion of ancestry that matches from these different groups across the genome, it infers your proportional ancestry based upon matching your genome to these reference population samples.
So we would consider the definition and consider at least anyone that has even, say, 99% European but 1% Black, that person do you consider admixed?
I would.
Just in terms of
Yeah, 1%. Yeah, that's right. Yeah, I would. Yeah, but in my model, I would. But they're going to cluster mostly with the European, but we can model that. So, what's the main difference actually between this and PCs? Reference and what? It's supervised. You're giving it reference, but you could do PCs. You could throw some. You could throw ancestral populations or reference populations in there, but a lot of times with the PCs, we don't know, it's not clear, for example, we don't know exactly what these PCs are reflecting, With these admixture proportion, we're making clear that we say that these are the 3 distinct ancestral populations and what proportion of ancestry do we have from these 3 groups. One of the drawbacks of these admixture methods is that the results can change if you say these people have 4 ancestral populations, So it's very sensitive to your assumptions on the number of ancestral populations, which is why a lot of people prefer using principal components, because you're getting all the genetic diversity and your captioning structure without specifying a lot of things you don't know. You don't know how many ancestral populations there are, and it may not be discrete like that. Like we say African, European, Native American, but there are subpopulations within those groups,
So is that the advantage of an admixture is some better power?
So the advantage, first, interpretability, right? Because a lot of PCs, we don't know what it is. At least for the admixture, we say, I find that mixture most helpful, particularly for continental ancestry. I can say, okay, this person is 30% European, African, but for subcontinental, you may not be able to capture that very well with this. Okay, so but for interpretability, it's fine, and we'll see actually that the proportional ancestry from admixture and the top PCs often correspond to each other. For admixed populations who have diverse ancestry, the top PC is typically reflecting continental ancestry differences. It matches quite nicely. So there's a nice relationship to them. But for interpretability, it's easy to interpret the admixture because this is the proportion from the different groups, but you have this issue that you're making strong assumptions of the number of underlying ancestral population, and so if you change that in your analysis, you can get very different results.
Anyone ever made a plot where they colored by admix? Like to do a PCA analysis, but then colored by the...
Yeah, I did that. I'll show you that in a minute. So here's an example for the HapMap AS. You're going to be analyzing this data. 150,000 SNPs, HapMap African Americans in Southwest, and the Mexicans in Los Angeles. You're also going to look at the European samples, Native American samples as well. And I think I put another in West African, so you're going to analyze that data. But anyways, I estimated admixture, supervised analysis, and this is what I get for the African Americans in MXL. So what are your thoughts on that? What are you seeing here? So for the autosomal markers, okay, you see, for example, African Americans in Southwest Texas are largely African-derived, some Native American ancestry, but European as well. But with the Mexican samples in HapMap, you got some Mexicans who have a lot of Native American ancestry, but you got some Mexicans who look almost 100% European. Okay, so a lot of variability even within Mexico. So my PhD student actually looked at this a little further and had found evidence of ancestry-related disorder in mating, where individuals who have more European ancestry within the MXL samples were more likely to mate with each other. Okay, people who have more Native American ancestry were more likely to mate with each other. So you have this assortative mating based on ancestry, which causes this population structure to persist because we're not mating randomly.
So in this study, you used 3 reference categories.
Yes.
And how did you, I mean, it's kind of obvious as you described the samples how you chose those reference categories, but you were just saying that there might be more.
Yes. So I gave it continental ancestry, okay? But there could be subcontinental that I'm not going to pick up with this admixture analysis. I may pick that up with the PCs. So here's an example, for example. So this is the proportional ancestry on average that I got. But here is, you probably can't see it too well, these are the PCs. So this is the PCR method where first, what is the first PC separating? Any thoughts on that? Okay. Okay, question.
So this is like the third time I've heard this idea about people not dating randomly.
Yes.
I'm wondering if you could break down that construct of what random means. Are you talking about geographically in terms of the randomness or lack thereof?
All the above. So people are going to mate with people who have similar ancestry that they have, right? So that's not random mating, right? So geographically, but also social, economic, ethnically. So people are mating at random. People are more likely to mate with somebody who has similar race. Yeah, SCS. Caucasian. Yeah, exactly. But here we're just talking about in genetic ancestry. Yeah. So first, what is the first PC telling us? So look at this one, because this is the method that we developed that takes into account relatedness. But what is the first PC telling us? Africans, everyone else. So separating the Africans from everyone else. And what is the PC2 telling us? European Native American. Okay. It's almost perfectly correlated with, the first 2 PCs are perfectly correlated with African and European. So you really need 2 PCs to really capture 3 dimensions of ancestry. Okay? And so just to show that these other methods actually run into some problems when you have relatedness, the first PC, for example, for the Eigensoft, the standard PCA, separates African versus European, but then it starts picking off pedigrees, okay? Because these people are related to each other, and so there are genetic relatedness, and so it's going to pick that up. Whereas we've kind of accounted for it, and so we're getting better estimates of ancestry.
Is there like a fit index, or are you just eyeballing it.
So when you say 50, so in here we kind of know the truth, right? Because we have reference samples and we're estimating it, right? But in reality, you're not going to know. So when you're doing the PC analysis you just did that Joey provided, he just did PCA. We don't know what those PCs means, but they are explaining some variation, okay?
And he had 3 of them.
Yes. So
Sorry.
4.
So you can tell I really.
Yeah, you did it.
So how did you get 4, Joey?
He's behind you.
Why didn't you have 5 or 3? What?
You have n item vectors. If you're just picking what to use.
He said, why only 4?
So if you look at a scree plot, right? Like when you do any other range variables, you can look at your scree, and as it gets further down the line, it doesn't matter anymore, right? Because it's not explaining variance. You can throw 10, the only thing you're hurting is your degrees of freedom. So I'm sure you get 4 because you were like, well, probably 1 or 2 that you
It's all already calculated. Most of the time, under calculation, what we pick up the genetic differences, the northwest, the southwest, and the Ashkenazi Jews. You really don't need, unless you have ethnic population, you really don't need that much PC to work with it.
We're not.
And it's also dependent on study. Some have 10, some use 6. I don't think there's a very definite answer that
There is not.
actually use 4, but it's a good
Well,
option.
you might look at like, okay, it might, 4 might explain like 90% of the variation. And you're like, all right, 90% is good.
No, it'll never be that much. It'll never be that much.
I don't know.
Yeah. Okay. Yeah. Yeah.
That might be a way that you can try that.
No, I mean, it depends. I mean, so like plots, scree plots, I often look at like the PC plots and like PC1, PC2, PC3, PC4. Once I just seen a bunch of clustering, a noise, and there's no clustering, then I know I'm done. And then a lot of times you guys look at those QQ plots. If you've seen some funky QQ plots, it could be because you're not adjusting for enough PCs. And so you might add more PCs and then redo it again. So people do all types of things with that. Paul?
Yeah, I was just going to reinforce the QQ thing. So there's a whole lot of variants that are related to ancestry. And if in your QQ plot, away from the really interesting cool hits at the end, along that kind of middle part, if you're seeing a big deviation where lots and lots and lots of variants are more statistically significant than you expect based on chance alone, this would be a definite place to look.
To start.
So you throw in another principal component and that the resulting QQ plot gets better, you've got your diagnosis and your cure at the same time.
Yeah, that's part of QC. Sorry.
I'm curious, I've heard about ancestry informative markers, and you had just mentioned doing the analysis with all of the SNPs in
Yep.
the data set. Can you talk a little bit about the choice of markers that go into
Yeah.
analysis and what is the preferred method or what are the pros and cons?
Yeah. So AIMS were used when we didn't have genome-wide data, right? But the problem with AIMS is that you really had to know more about the ancestry in your sample ahead of time. Like we don't know what AIMS, for example, are good for Native American ancestry. Okay, so they've done like analysis where they compared like using AIMS where they know that are really informative for ancestry versus using PCs, and PCs always outperforms it because PCs are giving you the AIMS in some sense. PCs identifies those variants that explain most of the differences that we see. So if you look at the weights that are given to the SNPs, those are actually that are going to get put out in the PCs are going to be the AIMS, and you have a lot more data for it as well. Question?
In terms of choosing the number of PCs and the relationship with the PC to your outcome,
Yeah.
should that inform how many PCs you choose or which PCs you choose, or is it really study specific?
So the purest statisticians say you need to choose that before looking at your outcome. It needs to be completely independent of your outcome. So everything I've done so far has been based only on genetic variation. Okay, so there are some people who do it based upon the outcome, but it should not be depending on the outcome, essentially. That's my point, yeah.
So if your sample size is small and you're trying to do everything you can to maximize your power, nonetheless, you shouldn't be doing that. It's not good.
I wouldn't. Yeah, I just wouldn't. Yeah, I'm not trying to lecture anybody, but I wouldn't do it. Yeah. Yeah.
Shouldn't you do more than one PC? You should do the initial PC to figure out what the ancestry looks like, right? And then you get
What do you mean you should use more than one PC?
Well, I mean, you do PCA twice. So, like, first you would do the PCA just to look at what it plots out to.
Yeah.
And then once you then use that as a QC of your data to take out like some outliers if you need to, then you rerun the PCA to get your eigenvectors, which you would then use as your covariates for.
I wouldn't run it again. Why do we need to run it again? We ran it. We're going to run it one time. We got a surrogate for Ancestry. That's what we're using. So you ran it one time. So typically that's what's done. You run it one time. You look to see which PCs may be informative for Ancestry or surrogates for Ancestry, and that's what you use as your covariates you adjust for.
You don't use it at all as QC if you don't know what the true background.
Some people do that. So for example, if you have one, let's say for example, everyone looks like they're clustering, but you got one really extreme individual. Some people use it to exclude, and then and if you're trying to do it as QC, then yes, you probably would do that as well. Yeah, if you're trying to identify like one outlier. But most times, for example, what we do, unless that sample is some like strange sample or we have like a strong justification to remove it, we include it and just adjust for it.
We almost always remove it.
Yeah. So what do you do? So who are you removing and why? Yeah, if it's something looks funky, yeah.
Yeah, and then we just do one. I mean, in theory, it should make a huge difference.
Yeah, because if you're adjusting for it, so I don't see why you really have to remove it unless it's really a bad quality sample. Yeah. So I know I'm running out of time. You sure? Okay. So anyways, this isn't about relatedness, but be careful for cryptic relatedness as well. Okay, so all these PC-based approaches that I was showing here, like if you don't take into account the relatedness in the sample, the PCs aren't reflecting population structure. It's just telling us that these people are correlated because they're in the same family. Okay, so just be careful about that. Everything we've done so far, these other methods, standard PCA assumes that people are unrelated, and any differences or similarities that we see is due to ancestry. But here, for example, we're just showing that even if you go up to 8 or 9 PCs with the standard approach, you're still picking up families in these studies. And one of the reasons why is that we had a paper that we published for HapMap just showing that we were able to infer the relatedness. And these people were supposed to be just trios, and we were able to recreate a 3-generational pedigree after we infer the relatedness. Okay, so just be careful. If you're doing these large studies, inherently there's going to be some cryptic relatedness that you're going to have to account for, and you don't want to use like Plink. There's other methods like linear mixed models that you want to use to account for the relatedness. So just beware. But this is essentially what we're seeing in these other methods, is that we're seeing these large families that were previously unknown, because when we did the PCs, we were trying to figure out, okay, this, because that's really motivated us to actually to develop this method, because we were seeing these crazy results that didn't reflect ancestry. Okay, and so just beware is that if you are seeing some funky results, you really have to investigate it and try to understand why. Okay, and it may not be due to ancestry. So I'll finish up with Hispanic Community Health Study. This is the largest study of Hispanics to date, looking at genetic risk factors for a variety of health conditions. We had 13,000 Hispanic Latino men and women who consented to be part of this study. And so we talked about, I showed 2-dimensional, but you can also do 3-dimensional plots, which is also sometimes really, really cool to show. Here, for example, we're separating, for example, the Puerto Ricans from the Cubans. And so and here is a plot based where we estimated proportional ancestry. So that's in one of the papers that we published. Here, we looked at proportional ancestry. We estimated continental ancestry. And so what's striking anyone of the Cuban, Dominicans, and Puerto Ricans versus Mexicans, Central Americans, South Americans? What's the big difference between those two groups? So Cuban, Dominican, Puerto Rican versus Mexican, Central American, and South American. Native American ancestry. So the Caribbean Hispanics have a lot less Native American ancestry compared to the Mexicans, Central, and South Americans. Okay, so when we say people are Hispanic, just know that just means they speak Spanish. That doesn't tell me anything. Yeah, exactly. Okay. So here, where I'm just showing that Puerto Ricans almost completely separate from the Mexicans and Central Americans. So that was a nice paper they published. But remember that paper, the figure that I showed you by John Novembre, where we looked at the European samples and saw that nice, so that the genetic variation mirrored from the PCs, mirrored geography. We did the same thing in Hispanics for individuals who both sets of grandparents came from the Central and South America, and so that there also was some nice clustering that mirrored geography well, even in these admixed samples as well. Okay, and so answer the question like would you use admixture proportions versus PCs? This is like PC, like these are like the later PCs, like this is PC like 3 and 4, and so that's capturing that. You're not going to get that so well with admixture proportions. Okay. Yeah. Okay. So we are actually showing that there is some geographic clustering that matches the PCs.
What's the map on the left?
On the left?
Yeah.
These are the top, the PCs, PC3 or PC4, and this is the map of the region. Okay, so it's similar to the map of the European PCA plot. And just showing that even in admixed populations, you are seeing similar patterns where geography actually matches somewhat, not as nice as in the November paper, but 1 and 2 are telling us about continental ancestry.
Oh, sorry.
Right? So, yeah. So 1 and 2 is really separating African and European, but then you got to go to get the more subtle structure, right? The later PCs are pulling that out. Somewhat country of origin for these individuals.
So the colors are random, right?
So, yeah, this is the color for the South America, and this is for this Central. Yeah. Okay, so let me just end by, I'm going to end with the next 2 slides, just to say that when we started doing the Hispanic community health study, we got a lot of pushback because our sample size was relatively small, only 13,000. Almost all the phenotypes that we looked at were previously studied in much larger samples, over 100,000 in Europeans. We found a number of novel genetic variants that you won't find in European samples. And so I think we have almost 50 publications that have been published or will be published soon where novel genetic variants have been identified in the diverse Hispanic populations that and some of them are Native American specific. So just showing that there is some value in doing these genetic studies in diverse populations. So I'm encouraged by the Alzheimer's disease community. There seems to be a greater push, at least recently, to start looking at genetic studies in diverse populations. So, Tim, is that your paper here, which is nice work. And then a novel genetic variant was found by looking at multiple ancestral populations, diverse samples together. It was published earlier this year. A lot of people here are authors on this, as well as this paper here. So I'm encouraged. So is anybody part of this ADSP, the Alzheimer's Disease Sequencing Project? So we have a few people there. Yeah. So we have data from Richard Mayu's group where we're looking at Caribbean Hispanics and families, and there are some potentially novel variants in these families that may be involved with AD that we're not finding in other populations. So there's a lot of value to looking at genetic studies in diverse populations, particularly for identifying novel phenotypic variants that may explain some of the health disparities that we see. So I want to plug some software. This is the Genesis software. A lot of the tools that I talked about are implemented in Genesis. If you're doing genetic association studies in diverse populations, check out the Genesis software. This is the link to it. It's in Bioconductor, and so it does principal components analysis and related samples, estimates admixture proportion, association testing, allowing for relatedness and population structure. So it's a tool set, but it does a lot of things that Plink cannot do. So Plink can handle some of the basic analysis. This can do some more advanced analysis, including like linear mixed models to account for correlation due to relatedness and population structure. So with that, let me just say that please go to the website that's here. and download the data. So we're going to be doing some principal components analysis and then mixture analysis with some genome screen data and trying to infer population structure. So for this afternoon, take some time to download the data at this website, and I think I'll stop here and happy to entertain any questions. Okay. Paul.
So on an ADS call, a certain project officer who will remain was comforted by the fact that there are some sites in Spain